Lesson 3.2, Organelles. We want to begin on the very outside of the cell with the plasma membrane. We're going to spend a lot of time in Chapter 4 discussing the structure and the function of the plasma membrane. It is composed of something called a phospholipid bilayer. The individual phospholipids are the little blue dots with the tails, and then there are a number of proteins embedded either in the surface or in some cases all the way through the plasma membrane. Again, we'll learn much more about its function in Chapter 4. This forms and maintains the boundary of the cell. We can use the analogy of the gatekeeper to describe the function of the plasma membrane. It controls all movement either into or out of the cell. If we move inside the plasma membrane into the cell, a number of different organelles and structures that we need to learn about. All the way normally in the middle is the nucleus. We can think of the nucleus as the cellular supervisor. This is where the genes are found, the DNA, which controls all cellular function. Uh, in eukaryotic cells, that nucleus is bound by a membrane, which is called the nuclear envelope. You can see in the diagram here in purple the nuclear envelope. The DNA is on the inside. That DNA exists in a form that is called chromatin. There is a dark structure inside the nucleus, which is the nucleolus. This is where ribosomes are formed. We'll talk about ribosomes in just a moment. There are also some openings called nuclear pores in the nuclear envelope. These are little openings in the nuclear envelope, and they are surrounded by proteins which control movement of material into and out of the nucleus. If we move outside the nucleus, the first organelle that we will discuss is the ribosome. The ribosome contains two subunits. These are called the large and the small subunit. Each of them is composed of some RNA and also some proteins. And these are assembled in the nucleolus, which we saw just a moment ago, that area, dark area inside the nucleus. The ribosome subunits are assembled in the nucleolus, and then they are transported out of the nuclear envelope into the cytoplasm of the cell. And when they are in the cytoplasm, they are the assembly line for protein synthesis. Any protein the cell needs to make, either enzymes or structural proteins, are formed on the ribosome. The ribosomes can be in a couple of places. Some of them are free out in the cytoplasm of the cell. Others are clustered on a membranous structure which is called the endoplasmic reticulum. The endoplasmic reticulum is part of something called the endomembrane system. Um, this is a series of membranous compartments inside the cell. There are specific functions and activities which occur in special environments inside the cell, and these little special environments are made by these membranous organelles. The endomembrane system includes the nuclear envelope, which we saw a moment ago. That's the boundary of the nucleus. And then if we move just outside the nuclear envelope, we will see the ER, the endoplasmic reticulum, and then the Golgi apparatus. And then lastly, there are a number of transport vesicles that break away from the Golgi apparatus. And so we'll discuss each of these components of the endomembrane system individually, and then we'll look at a diagram that puts it all together. If you look at the diagram in the center, we see that generalized cell. You see the nucleus that's dark purple there, the nuclear envelope, and then just outside the nuclear envelope, there are some folds of membrane. This is the endoplasmic reticulum. Actually, there are two parts to the ER. Part of this is called the rough ER, because it appears rough. It appears rough because of the ribosomes that are embedded in the ER. So if you look at the larger diagram, you see these folds of membrane and the little yellow dots represent the ribosomes, which are where proteins are being synthesized. As those proteins are synthesized on the ribosome, they pass into the folds of the ER. Uh, Here, these proteins can fold up into their functional shape, and also some modifications can take place to those proteins. There are other portions of the ER that lack ribosomes, and this is called the smooth ER. The smooth ER is involved in lipid synthesis inside the cell. Um, so we can think of the ER as the manufacturing plant. If the ribosomes are the assembly line, then those are housed, many of them, in the ER, where a number of things, both proteins and lipids, are synthesized. The Golgi apparatus is the next portion of the endomembrane system. We can think of this as the packaging and distribution center. Proteins, many of them are assembled on ribosomes inside the ER, and then there are little vesicles, little spheres of membrane that break away from the ER. They travel to the Golgi apparatus, and those proteins then can be packaged and they can be uh, shipped to different destinations in the cell. So these new proteins can be modified in the ER, they're going to be sorted, and they're going to be packaged. All of these proteins, structural proteins or enzymes that are manufactured in the cell, have a specific destination. They have to arrive at a specific place inside the cell where they're going to do their job. And the Golgi apparatus is involved in getting these proteins to the correct location in the cell. So this diagram shows the endomembrane system. Again, if we look down at the very bottom, you see the nucleus. Extending out from the nuclear envelope is the endoplasmic reticulum. Again, the little yellow dots are the ribosomes. So we're going to build some proteins on those ribosomes. And then you see these little transport vesicles. These are little shuttles, little spheres of membrane. They break off of the ER. The little blue structures represent brand new proteins that have just been synthesized, and these transport vesicles then move to the Golgi apparatus. They fuse with the Golgi apparatus. Those proteins can be modified, sort, packaged, and then sent on their way. If you look towards the upper right-hand portion of the diagram, this represents some proteins that need to be secreted. Many things that are manufactured inside the cell need to be released outside of the cell. And so these secretory vesicles break away from the Golgi. They move to the plasma membrane, they fuse with the plasma membrane, and then those proteins are going to be released on the outside. On the left-hand side, there are some little transport vesicles that may break off of the Golgi, and they fuse with something called a lysosome. We'll discuss the lysosome in just a moment. Lysosome breaks down cellular debris, or sometimes incoming material can be broken down in a lysosome. But those digestive enzymes that have to be in the lysosome are manufactured on a ribosome. They pass from the ER to the Golgi. The Golgi sorts these specifically to the lysosome. You can think of the lysosome as the garbage disposal, I guess, of the cell. Again, a lot of things, byproducts, debris inside the cell needs to be broken down and disposed of. And so there are digestive enzymes inside the cell that can break down these waste products. Certainly, those need to be contained. So mentioned earlier that different functions of the cell have to be contained in specific compartments. This is another one of those where we need to contain and compartmentalize these digestive enzymes. Um, This is the way some of our white blood cells work. Whenever some of our white blood cells engulf something like a bacteria, it comes into the cell in a little vesicle. It can fuse with a lysosome, and then those lysosomal enzymes can destroy that bacteria. There are a number of diseases that result from uh, dysfunctional lysosomal enzymes. One example is called Tay-Sachs syndrome. There are some lysosomal enzymes that do not function, and as a result, some of these toxic byproducts can accumulate inside the cell and eventually kill the cell. Another membranous compartment or vesicle that's similar to a lysosome is called a peroxisome. There are some specific reactions that occur in peroxisome, and they result in the production of hydrogen peroxide. Hydrogen peroxide would kill cells very, very quickly, and so it has to be contained inside these peroxisomes. And then there's another enzyme called catalase that can break down that toxic hydrogen peroxide. uh, Two energy-related organelles, and we'll learn much more about these in some of the later lessons when we get into cellular respiration and then into photosynthesis. The mitochondria. The mitochondria, everyone has heard called the power plant of the cell. This is where the majority of the ATP is produced inside a cell. If you look at the diagram, they look kind of like little sausages. The mitochondria has two layers of membrane. It has an outer layer, and then the inner membrane of the mitochondria has some folds in it, forms uh, some areas where the enzymes that actually are involved in ATP synthesis are found. And the chloroplast is the site of photosynthesis. Again, we'll learn much more about this in a later lesson when we specifically look at photosynthesis. Um, the chloroplast also has two layers of membrane, an inner membrane and outer membrane. Inside, there are some stacks called grana that look kind of like stacked up coins. And this is where the chlorophyll is found, which captures sunlight and drives photosynthesis.